Hi, my name is Chris O'Donnell and this is my presentation for the tissue engineering poster for the investigation of TCP on gelatin scaffolds. The main objectives of this investigation are to fabricate TCP and gelatin pristine scaffolds using a freeze drying technique and test mechanical properties, degradation properties, cell viability and cell proliferation. The labs were conducted over 4 weeks, and in the first lab we created the TCP and gelatin scaffolds using freeze drying technique, which had a 26-hour freeze drying cycle. The second lab was the compression test and the degradation study of gun. The third lab was the degradation study results, and the SEM was used to obtain images of the microstructure of the surface, and the final lab was the hoist array used to correlate fluorescence and DNA concentration, which correlates to cell numbers. So the compression test showed that the TCP in the blue up here was much stiffer and had a much bigger spread from this box plot of stiffness when compared to the gelatin only scaffolds. The mean was about 0.08 megapascals for the TCP and 0.03 megapascals for the gelatin only. The degradation study was carried over 7 days and it actually showed no degradation. This is likely due to the amount of GTA used in production of the gelatin scaffolds at the beginning. This is shown from literature to improve the amount of cross-linking, which can mean that degradation occurs much more slowly. The SEM showed porosity for the degradation for the TCP and shows no porosity for the gelatin only. And this is shown here. There's a 44% porosity for the TCP and 0 for the gelatin, along with 350 micron average diameter compared to 0 for the gelatin. The DNA fluorescence test from the hoist array shows a nearly 1 regression between the fluorescence and the DNA, which means this is a really accurate model. And then this was used to calculate that there are 9 million cells proliferating from 1 million seeded cells for the TCP and about 4 million cells proliferating from the 1 million for the gelatin. So concluding now, all the results line up with our hypothesis, which means we can take our hypothesis as word, and bar the degradation study, which shows no degradation occurred, so possibly more time would be needed for this. It is shown that TCP enhances the mechanical effects, cell proliferation and cell viability of gelatin scaffolds, and I would like to thank Manus Biggs, Dave, Maya and all the lab technicians for their help throughout this lab.